Assalamualaikum and hi, my name is Muhammad AIMAN bin Bahari, number matrix E1A0523.
Hi, Assalamualaikum. My name is Muhammad AZAMUDDIN bin SALLEH. My matric number is E21A0531.
Hi, my name is FARHANA. My number matric E21A0490.
Hello, my name is Fatin Nurshafiqah and my matric number is E21A0751.
Hello, my name is ERINA Binti HATA. My matric number E21A0472.
My name is Binti JUNOH alias Muhammad JUNOH and my matric number is E21A0611.
Transgenic technology allows the introduction of the new functional genetic material into the germ-line. The first advised eukaryote to prove to such genetic modification were mouse. In 1980, Gordon and Rodel was invented the transgenic technology. There are three approaches to genetic transformation of the mammalians. Germ-line are currently used and also past 20 years. These are DNA microinjection, retrovirus mediated gene transfer, and embryonic stem cell mediated gene transfer. Important features of all gene transfer approaches are their efficiency, the character of foreign DNA processing during the transfer process, the integration mechanism itself, and the characteristic of donor gene expression. The first one, DNA microinjection, was the first transgenic technique demonstration to be effective in mammal. With these approaches, one cell fertilized egg are microinjection with purified DNA, and surviving embryos are then re-implanted into the uteri of females that are hormonally and psychogenically competent to carry a pregnancy. The last one is embryonic stem cell mediated gene transfer. The blastocyst is harvested and mixed with recombinant DNA and inserted back in blastocyst. In this technique, DNA was mixed with sperm cell before in vitro. 30% of offspring mouse were integrated in foreign DNA. The basic principle of sperm-mediated gene transfer is seminal plasma-free. Sperm cells are suspended in the appropriate medium and then incubated with DNA. The result DNA carrying sperm are then used to fertilize eggs via in vitro fertilization or artificial. This method introducing the transgenic DNA directly into the zygote at an early stage of development. The second one is retrovirus mediated gene transfer. In this method, retroviruses are used as vector to transfer genetic material into the host cell, resulting in a chimera, an organism consisting of tissue of part of the diverse genetic constitution. When the cells are infected by retrovirus, the resultant viral DNA, after reverse transcription and integration, become part of the host cell genome to be maintained for the life of the host cell.
Next, we will be talking about application of transgenic technology in animals and plants. Technological breakthroughs in the efficiency of transgenic technology have significant medical implications. The production of therapeutic proteins in animal bioreactors is well established. Therapeutic proteins are used to treat patients suffering from cancers, heart attacks, strokes, cystic fibrosis, diabetes, anemia, and hemophilia. These proteins are produced by using microbial fermentation on cell cultures in transgenic animals and in transgenic plants. And the first products are starting to reach finalization. Pigs are example of one bioreactor animals that have been now being genetically modified to improve their suitability as organ donors for xenotransplantation. But many challenge remains. Bioreactor animal can be detected by their ability to express transgenes in milk-producing animals. In specific, has led to development of bioreactors or animals that produce large amounts of a given recombinant protein in their milk in fully biologically active forms, such as through proper post-translational modification (PTM) for purification and therapeutic use. While xenotransplantation is any procedure that involves the transplantation, implantation, or infusion into a human body, either live cells, tissues, or organs from a non-human animal sources. So, transgenesis in conjunction with RNA interference to knock down gene expressions has been proposed as a method for making animals resistant to viral diseases, potentially decreasing the possibility of transmission to humans. Next part, we are going to talk about advantage of the transgenic technology. Firstly, it is help to improvement in insect and diseases resistance. Every year, insect and diseases attack causes significant yield losses in crop plants. Furthermore, insecticides and pesticides used to control insects and diseases are expensive and have adverse effects on other beneficial organisms, such as parasites and predators. Insect-resistant cultivars in several crops have benefited greatly from gene technology. Cotton bollworm resistant cultivars, for example, have been developed by transferring a gene from the soil, Bacterium bacillus to Greengiensis, into cotton plants. This results in significant cost savings for insecticides. Furthermore, the technology is eco-friendly. Next, it's help for improvement in quality. Quality can be determined in three ways: nutritional quality, market quality, and industrial quality. Gene technology has greatly helped in the improved performance of all three types of quality in various crops. For example, gene technology has enabled tomato oxidation and softening to be delayed, resulting in safer transport and longer storage. This was accomplished by modifying the genes that code for the enzymes responsible for maturity, HAZLIN forming enzymes EFE, and softening polygalactonase. Other quality enhancements include non-browning potatoes, wet flour starch composition, and increased oil content in oilseed crops. Next, it gives an advantage to industrial products. Transgenic plants have enormous potential for producing biodegradable plastics, therapeutic proteins, pharmaceuticals, and edible vaccines. It could also aid in the production of biodiesel or petroleum products. And the final advantage that we can be discussed is about rapid and accurate technique. Gene technology is a fast and accurate method of crop improvement. This technique takes 4 to 5 years to develop a cultivar compared to 10 to 2 years for the conventional hybridization method. Furthermore, this is a highly reliable technology. For the disadvantage of the transgenic technology part, we can see the obvious disadvantage that can be seen. Firstly, it raises the cost of cultivation and makes farming more susceptible to marketization, which results in immoral profits. Next, transgenic crops endanger not only farmers but also commerce and the environment. It has been biologically altered. As a result, biotech foods may endanger human health. And lastly, excessive production of genetically modified foods will eventually render them ineffective because the pests that these toxins are used to deter may develop resistance to them.
There is of transgenic technology in the field of fishery, especially fishery cultivation, is intended to improve the quality of fish cultivation. In education, transgenics are performed to obtain the desired properties and increase product. Zhu et al. Report that they have been able to produce transgenic fish by transferring growth genes. They have successfully created transgenic lock, goldfish and goldfish using a red metallocyanine promote inoculated with the GH gene structure of transgenic fish turn out to be 3 times less than control fish. In education, food product touch gene engineering known to affect health. Human because it transfers gene from other bad organisms in food to give new characters to the food. Because genes interact with each other, new genes are inserted at random. This can interfere with the normal function of plant genes. This leads to unpredictable effects which may not be normal for plants. The most unpredictable effects on human eating habits, such is the product of new toxins and algorithms.
Field applications of transgenic technology, transgenic animals, particularly mice, have been almost entirely used for basic research on mammalian developmental genetics. This technology, however, has enormous potential for livestock genetic engineering. Many applications of transgenic mice for baseline studies have previously been investigated. The use of gene insertion to elucidate gene regulation mechanism will be reviewed briefly in this study. For example, hepatitis B infections. These examples are discussed only because they demonstrate several ways in which transgenic experiments can affect laboratory animal management. Gene regulation and transgenic mice transgenic technology has made an important contribution to biomedical science by determining the mechanisms by which genes become differentially regulated during mammalian development. It is critical to understand that during the differentiation process, tissue-specific genes are activated while most others are permanently silenced. Lines must be created and maintained aggressively because the presence and expressions of transgenes can affect fertility. It is critical to breed transgenic aggressively until lines are formed. To summarize, transgenic technology is one of the most significant scientific advances in recent genetic development history. It conducts numerous studies, particularly in disease prevention and livestock genetic modifications.
A transgenic organism, also recognized as a genetically modified organism, is one that has been altered using recombinant DNA technology, which involves combined DNA from different genes or inserting DNA to a new gene. Method from University of Malaya, how making a transgenic plants. First, design and making the gene construct. When create the transgenic plant, you create a construct which contain the gene you are interested in, the gene you want to test, and then it needs a promoter which effectively drives the gene by crossing to the produce messenger RNA, which is then converted to protein. So, need a gene, a promoter, and a terminator, which will determine when the gene stops transcribing. Apart from that, we add antibiotic-resistant gene to make it easy to identify plants that have been transformed properly. Second, introduce transgene to plants using agrobacterium. To use agrobacterium to change leaf, we must first change our agrobacterium which the gene of interest combined with the biotic-resistant gene. Then we take our leaf piece, damage them by cutting them, for example, and introduce agrobacterium to the cut leaf piece. We only left it for about 10 minutes, which gives the agrobacterium enough time to integrate its DNA into the exposed cell on the leaf surface. After that, we remove the left fragments, let it recover, and then starting the plant attempt to on media contain antibiotics. Next, growing the transgenic plants. If the agrobacterium is successfully in change part of the leaf, it affects the individual cells in the leaf. These cells will multiply and divide. Plants have the incredible ability to effectively reproduce themselves, which is known as totipotency, and they can create an entire plant from just one cell. Next, confirming the plants are transgenic. We now have almost the entire DNA sequence of a plant because we have the entire gene. As a result, we can use relatively simple procedure known as PCR, polymerase chain reaction, to determine when our transgenes are located in a plant gene. This process can define how many times the transgene has been integrated into a genome. It could be multiple times, for example, and need to know because it could affect plant performance. Thus, what transgenic can tell us, the real benefit of using plant transformation transgenic is that we can study individual genes in plants in detail. We can demonstrate the function of a single gene. We can look at the tree and see how it grows, how effectively it grows, and how tall and large it grows, and predict what will happen in different situations, such as in the garden. Most importantly, we can see how it flowers and how many fruits it will process.
This is a reference that we use to find information about transgenic technology.